Good afternoon, everyone. Oh, I didn't test that. I will go turn this on right after my intro. Welcome to Live at Lunch. This is a free noon hour presentation that happens on the third Wednesday of every month, and it features staff from the museum as well as researchers and fascinating community members. My name is Kim Goff. I'm a learning program developer here at the Royal BC Museum, and I'd like to acknowledge that we are on the territory of the Lekwungen, known today as the Songhees and Wisepsum First Nations. Lekwungen means place to smoke herring, and we're all very honored to be able to work and to live on this beautiful territory. Today, I'm happy to welcome our curator of entomology, Dr. Joel Gibson. Joel grew up in southern Ontario, and after completing degrees in wildlife biology and insect systematics, he was briefly a high school teacher. That would drive anyone back to school. Probably he returned to research and earned his doctorate degree, which took him around the world studying and collecting insects. He joined the Royal BC Museum in 2016, where he has worked to expand and share the knowledge of insect biodiversity of our province. Please welcome Joel.
Thank you, Kim and Jenny and the whole crew for including me in this series. I think it's a great thing that we do at the museum, and I like the fact that it's a priority for us to sort of just chat with the community, and I like to think of this as sort of a conversation back and forth, even though I'm doing most of the talking to start with. But, you know, we can try to leave some time at the end if we just want to chat and talk about other insect-related things. But the big focus is sort of in my mind when I was talking with Kim about putting this together is like just an update, like what have we been doing lately with the big focus on the research part. I know there's a lot of discussion in the past and in the present and in the future about the new building and new collection spaces, and there's going to be a lot of great discussion about that, but I'm not going to talk about that now. I'm happy to talk about the end, but now I'm like, no, here's the research we've been doing, even though we're still packing, and the research that goes on, and a little bit with the new building, one of the things we have in mind is it's going to be a great chance to continue this. And the other sort of point I'll keep coming back to is none of this is all me. A lot of it is none of me. This is a lot of other people. We collaborate with a lot of groups outside of the museum and the different departments within the museum. So it's insect research, but it includes a lot of other people. It's a lot of preamble. We did territorial acknowledgement, which I think is important. And so the context here is every time, I like to put this slide in every time I give a talk because it reminds me, okay, what is my job as the curator? And it's always these three things: collections, research, outreach. The funny thing being, there's almost nothing I do that isn't all three of them. Like, this is technically outreach. I am reaching out to the community to share what we do about research, but none of the research we do could be done without the collections themselves. So I don't know which one this is most important of the three. Maybe it's all three. But a little background, just because I always like to reset it in case I get to update any of these numbers, and I did update these numbers if you've ever asked before how many things are in the insect collection. And the entomology collection now, as of the last time I checked the database, and we have our new database and updating, it's about 460,000 records. So those are individual pins or vials of things. Now, the actual fact is there's probably well over 1 million. There's probably that many as well that don't have a number on them yet. 460,000 is just the ones that have started to be entered into the database. And we do record things by individual insects. So we always say we're the biggest collection in the museum because there's no way they have 460,000 paintings. But then archives will point out and be like, well, if you consider page by page, they have millions and millions of pages. So I guess we're not the biggest collection if you look at it that way. It is a focus on BC and the Yukon, which is not uncommon for most of our collections. And as I said, we are working, even though the packing and moving is biting into that. One of the big goals we have is to make it as accessible as possible. And that's why I always emphasize how many things are in the database, because the point is the database to be public. It's publicly available. It can be accessed by anyone. And we're working with how to make that more user-friendly, more able for people to get the data out of it. If you are curious and if you're the type of person that likes to rank things, What do we have the most of in the insect collection? It's still beetles for now, even though I work mostly on flies, and I've been here for 10 years, and I haven't quite gotten there to be more flies than beetles. But it is pretty equal for the bigger groups. One thing that's always interesting is we have way more spiders and dragonflies than most collections do by proportion, but that's because we happen to have great researchers and teams working on spiders and dragonflies. So that always happens at collections. You have most of what people like to work on. Now, looking at some of these raw numbers, I'm going to keep with that theme of, okay, why would you put so much work going into the database? There's other things you can do. And this was a question that has been kind of rattling around in my head for a while, and I ran into a colleague from the University of Northern British Columbia and talking to him about it. And frankly, this was also sort of a product of sort of post-COVID recovery and how can we do things remotely. And he had a group of graduate students that he was working with in Prince George. And he said, they want to get collection and data experience. And we're like, well, we're still recovering from COVID and it's expensive. We can't have all 5 of them come and live and work in the museum. So we were like, okay, what can we do remotely with the data we had? And we also had the opportunity that I could help co-teach a class with them. So him and I taught a class together talking about kind of principles of curating insects and how do you organize them and what kind of data do you get out of them and why is it important and ethical concerns that go into collecting and things like that. And the course was amazing. It was an incredible group of students. And we said, okay, well, we have to have some way to evaluate. As a teacher, I'm like, you have to have a mark. You have to be able to say the students did something at the end. So we said, fine. Each of the students do some kind of analysis on a data set that we have and what can you get out of it. And they did such an amazing job. We're like, okay, we're going to publish this. Like, this is way too good for us to just call it a term paper because it was 5 term papers that were incredibly well done. So we put them together and published them. And one of the interesting things to come out of this paper was, and we emphasized this a lot at the end. We kind of got the chance to wave our hands and proclaim for what we thought is important, is this was 5 students that never stepped foot in the collection, but did this kind of analysis. And not to say we don't need to go in collections, not at all what we were concluding, but just the volume that you can do even without that. Imagine how much more you could do if they had have been able to. So what they did was they took 35,000 records, and like I said, dragonflies are an emphasis, and they have been for decades, so we have really, really good dragonfly records, and most of them have already been put into the database. So this was a good chunk of data. I could send them 35,000 records of dragonflies and say, what can you do with this? And they did an incredibly large amount. And this is what was published. I mean, it's publicly available if you want to look up that paper. We're very proud of it. And the amount of things that these students did with the data is incredible. So looking at the 35,000 records, one of them, she did this and said, well, 99% of them are identified to species, which is incredible. Most of our collection isn't identified that well. And almost all of them have complete location data. Almost all of them have the date they were recorded and who identified them. So this is just kind of a neat, I just like the look of it because it made me think of like a 1970s middle school. This just looks like the art that was on the wall of my cafeteria when I went to school. So when I saw that. I guess in the 70s, everyone wanted schools to look like this, so it affected me. So just looking at the completeness of the data is like, wow, that's a great set of data we have. But then what do you do with a really nice complete data set, 35,000 points where you collected a dragonfly in the province? Well, you can do things like this, which this is a heat map where if you take all of the records from a location, and the more they overlap, it's stronger color. So if you actually look at it, something that's red or yellow, there's a whole bunch of points in a small area. And then purple means a few, and white means basically none. So this is a way of visual. I mean, you could just as well make these points overlapping, but this sort of emphasizes areas a little bit more than that. And you get interesting conclusions like, Victoria gets to be closer to yellow and red, and the Okanagan Valley, which that makes sense. Victoria is here. We can collect a lot here. Okanagan Valley, because one of the leaders of our dragonfly research, Rob Cannings, who's here, grew up in the Okanagan Valley and would go back there and do a lot of collecting there. So that is a spot where we have a lot of records from those 2 places. Other things, those of you familiar with BC, what else do you see on this map? Is you can see the outlines of roads. And it's funny, no matter which collections you look across, you end up having these lines where most of the records are along roads because that's where you can get to. And you get unusual things, like there's huge swaths of the province that just are all white, and those are white for hundreds of kilometers around it, which means as good of records as we have of BC, there's still big stretches like we don't know what dragonflies are there because we don't have any records from it. So this is nice, where if you take what to me was just a nice list of dragonfly data, put it in the hands of this student, and she was much better at data visualization and being able to put this together. So other things you can do looking at it historically, and this was an important point for me to dwell on as well, is if you look at who was it that did the work on these. Now the people that collected them, there's a lot, going from 1913 up until I think 2010 was the most recent ones. There's a lot of people that collected dragonflies, that's great. But if you also look at who was it that identified all of these dragonflies, it ends up being somewhere around 90% of them were 5 people. Now, yes, 3 of them were brothers, which is interesting to note. The other thing I noted was that all 5 of them are also currently retired. You're like, oh, what does that mean? That you're like 95% of your effort in one group is amongst people that worked very hard but are no longer working full-time at it. They're retired. So that was another thing we emphasized and sort of tied it back into the fact, okay, these are students that need to get training, and we need to be training more generations of people to do it, because we have this odd phenomenon where a lot of the expertise, specifically in specimen identification, and even to some degree like collection management and database management, is retiring out. And are we making sure that that knowledge is being passed to another generation? And this was a bit of a stark reminder of it, that here, 5 out of 5 of the top identifiers are no longer employed to do that. Other interesting, and this was sort of next level computer expertise that I didn't have, but one of the students, what she was able to do is to just take a big spreadsheet, which is 35,000 records of specimens, and she did text data mining, which if you're familiar with is being able to just look through and just look for words. Doesn't matter where they are, doesn't matter what context they're in, can you find the word somewhere in the document? And what she did is she was looking for specific words related to habitat. Like, is it a wetland? Does it have bulrushes around it? Is there something related to temperature or weather? Which all of the different records have different amounts. Nobody records their data the same way. But when she did that, she was able to pull out a lot more information and be able to parse it together. Now, it wasn't all of them, but like 1,600 of them actually had really detailed habitat data, which is interesting when you consider like this wasn't necessarily a study to go and study the different habitats and wetlands of the area. It was just to look at the dragonflies. But as a side effect, you ended up with all of this habitat data. And that is interesting when you look at a lot of databases. If you look for all the other words that people didn't intend, you can start to pull out all this other very useful data. And then she got to draw this really neat Venn diagram showing how all the different types of descriptions overlap with one another. And then eco-province, this is something that we've been doing a lot, and it'll come up again in a minute, is just the idea if you were to group all of those habitats in the province into rough areas. And one way that's typically done with British Columbia is these 10 eco-provinces. You know, there's the coast, and then there's the Salish Sea, also called the Georgia Depression, and then the interior divided into sort of southern mountains, southern central mountains, central. And if you were to take all of the dots and group them together, then you can say, okay, every one of them, even though no one ever goes out and collects and says, I've got this dragonfly from Clinton, and I know this is the central interior mountains. Nobody writes that down. They write down Clinton, and then they put it in the collection and they move on. But what she was able to do is just take every location, use a different list, and add the eco-province label on them, and then analyze every specimen from an eco-province. And you can do things like this, and that is a complicated statistical analysis on the side, where if you take all of the species in any given area, how similar are eco-provinces to each other? Not do they all have the same exact species, but do they have sort of similar groups of species? And you can end up with these interesting groupings, which we've actually found in other groups that have the same, where you end up with the Georgia Depression, the coast and mountains, and what's the other one? The southern interior. So the south and the coast is one group of communities, and then the interior and the north is a different group of communities. And there's no one given species you can say that. I mean, there's some that are only in one place or not another, but if you look at a group of species together, you end up with this divide. And other studies have kind of found similar things with other groups, that you end up with 2 larger regions of the province, or 10 regions, or hundreds of individual locations. And it's nice when you can dial in and out to be what level do you want to look at it. Another one, and this one I thought was particularly cool. Another thing people don't know, and that's really, don't worry about trying to read that in detail. Don't break your eyes. But another thing, people sometimes will record the elevation they're at. I mean, people that are particularly interested in doing alpine things, of course, will, but I don't always think of it. So I just put down the latitude and longitude. I can get that off of my phone, and you move on. But if you go on Google Earth and you put in a latitude and a longitude, it'll tell you what the elevation is at that point. So what she did is she just took every location, put them into Google Earth, and then had it return an elevation for it. So then she put it back into the database and said, oh, I just added elevation for, 55% of the records, most of which had no elevation recorded. And then you're like, oh, is there a difference with different species at different elevations? And she did find things. And this was what she did, was she grouped them by genus, like not the individual species, but different genera of dragonflies. And she found some of them are never found above 500 meters. Some of them are only 10% of the time found below 700 meters. And you're like, oh, you never could have asked that if you didn't know what the elevation was, and you wouldn't have known the elevation if she hadn't done this secondary thing to add it. And just going back to the point, none of these women ever went, and they were all 5 women, none of them stepped foot in the collection. They did this remotely. And so our conclusion from the paper was, if you have good collections and you can get as much of it into a database as possible and turn it loose to the public and the world, they can do these great analysis with it. Because over the last 100 years, the work of collecting these specimens has already happened. There's a lot of work going into identifying them, and there's always work needed to be done to identify more, but the analysis can be done. So we left it very, very strongly word at the end of the paper of like, you have to put money into databasing things. Like, that is the real accessibility of this data. And it doesn't have to be just the specimen records. I mean, there are iNaturalist records, and people bring that off to like, what about photo records? And one of the things we had found is actually the last 10 years, there's a lot less dragonfly records. And you could say, well, Joel, that's when you've been working here. You're terrible at your job. That's when you stopped collecting dragonflies. And I'd say, no, it's not my fault. But also in the last 10 years, it's become a lot more common to collect dragonflies photographically. And dragonflies are particularly good at it because they're a lot easier to identify from photographs. Butterflies are good for this. Dragonflies, some bees are very good for it. Other insects don't lend themselves so well to it. They're too hard to identify from photographs, but dragonflies are. So you could say, well, fine, if you look at the 29,000 records on iNaturalist, you could do the exact same analysis we just did with that, and now you're just using photo records in addition to museum collections, and now you have a longer time span. And as you point out, the 29,000 records in the last 10 years tend to be sometimes in unusual places people don't often get to. So these are all wonderful things that we've been thinking of. Not as applied and not as collaborative outside. So I'll bring up another example. I'm just going to keep firing examples of studies. This was a paper that was the product of Agriculture and Agri-Food Canada. They said we have a new pest. Quick ID on that one. I mean, if you're reading it, you'll know that's not a wasp. That's a moth that has clear wings, makes it look like a moth. Really good mimic. Even flying around sometimes, you'll even swear they're buzzing and flying like it, and then they land and it's a moth, not a wasp. The other thing, and it's probably defensive, it keeps them from being eaten by birds, but some of them can be pretty important pests. And this came to me, Tyler, who works at Agriculture Canada in Summerland, was like, oh, one of these is a threat to apple trees and cherry trees. They kind of get under the bark. They can, really damage the trees, lower fruit output, big problem. They want to not have this threaten apples and cherries in the Okanagan. So they said, okay, well, can we look into ways to monitor it, control it, hopefully not pesticides, but if that's one way to deal with it, and there's other methods they can, But the question is ahead is, we don't know how many other types of this moth there are in the province. How many other wasp-looking moths are there? We didn't know. So we're like, okay, let's dive into the collection. We'll look at collections all over the place. And we do something like this, which is just a checklist of the clear-wing moths of BC and the Yukon. Turned out there was 22 species. 5 of them are introduced, most of those pests. 17 of them are native. And yeah, they're on different plants, but probably not a big pest. So wiping out all the clear-wing moths probably would have been a bad idea, because there's 17 of them that aren't threatening any of your agriculture. But at the same time, we were also to pull out, okay, what records do we have of what plants are they on? And at the same time, well, fine, we'll put it in this eco-province approach, and how many are in each of the different regions. And I was surprised to find there's 7 species of them in the Yukon, which I wouldn't have guessed there were that many up there, but there are. And so this approach is something we commonly do in the insect collection, because it's easy and it lends itself, it's not easy, it's a lot of work, but it lends itself to what we have. We have collection data, there's a place, and there's a species name. What do you do with that data? So we do checklists. We did one for the stiletto flies of British Columbia and Alaska. And this one, similar, 34 species. We had about 2,100 specimens. And this one, we knew that we had collected a lot here over the last 50 years, but we knew there were records from other places. So we would get specimens from Alaska. We got some from California, even some from Ontario, people that had come here at some point to BC and collected a few. Give us your records and put these together. And this is great, because this is a group of flies, stiletto flies, just because they kind of look like a little flying knife. I don't entirely know the name on that one. It's a bit of an odd common name. And they tend to be in pretty dry areas, sometimes in sand. Like the females will sort of just bury eggs in the sand. And they're predaceous. They'll grab other insects out of the air and eat them. The larvae probably are predaceous as well. We don't know a lot about the larvae. But you can do this. But this is another group that there's not that many people that study them. One of the motivators for this is the person who had done the world catalog of this group didn't have many records from BC, and he hadn't really included a lot of records from BC. So his records were very spotty, and was old and retired. He just wasn't able to work on it anymore. And we're like, okay, can we finish this and do the BC part of it while we can still ask you questions and do things? So some of this is motivated by urgency of time, but we have the data. Let's use it. And this is when it's just us. It was Rob and I did this, and we're in-house, and it's great to do. Rob Cannings has also done a bunch of other neat things, which I haven't got to do, I'm really hoping to, with fossils. He's done some fossil work lately, which is the other huge benefit of working at a museum like this, is we have a paleontology department. We have people we can ask, like, what does this fossil mean? How old are these rocks? What does this mean? And Rob has been doing a lot of that and just coming up with really neat, interesting things. So entirely new groups of dragonflies that are kind of intermediate between damselflies and dragonflies, based on these pressed fossils that are perfect encapsulation of the wings, and you can compare them in shape of the head and be like, yeah, this doesn't compare to either of them. One particularly neat one, Rob brought this in a little box. This is a head of a robber fly that was found in a midden of prairie dogs. Yeah, prairie dog midden, which was, 10,000 years old. And you realize these things can be very old. Prairie dogs will just bury things and then die, and 10,000 years later, everything they buried is still there. So it's technically considered a fossil because it's 10,000 years old, but Rob was still able to identify it to what group of robber flies it would match. So this is an odd case of somewhere in between working with mammalogy, the mammal folks, and working with paleontology, but it's also insects, and the help of Hugh in the basement, who had this incredible camera set up, Hugh McIntosh, that was able to take these photographs. So this is a great collaborative effort throughout the museum for something that kind of can only uniquely be done here. Now, all of this, and I'm very proud of myself, halfway through it, I haven't mentioned shoreline flies once. But now I'll spend the rest of the time, because that's what I like to work on the most, is shoreline flies. So I can just turn it over to that. And there's good reason to. I always give the justification. It's because BC has more coastline than the United Kingdom. BC has more coastline than India. we just have a lot of beach. And yes, it's because there's all the tiny islands and the distance around each island counts. But 6,000 islands, 27,000 kilometers of coastline. And when I first moved here and started working, the insects on those coastline had never really been a focus. I mean, it also applies if you extend it down up to Alaska and down to California. There haven't been a lot of people that focus specifically on shoreline insects. We have a long talk about that, and Henry Chung and I do about, well, because people just think there are no such thing as ocean insects. That's why they never did it, and we're finding out that's wrong. So one of the first things Henry and I did when we'd go out and sample on these beaches was we were looking at barnacles. Because I found large adult flies crawling around on barnacles. Henry was able to help me identify the barnacles. He was able to collect them. So he could scrape the barnacles off the rock. I would sweep my net around and collect the flies. We'd both come back to the museum and try to see what we found. I found these wonderful flies that I could identify. He pried open the barnacles and found there's things living inside the barnacles. And we were able to put this together and realize one of them we knew. These are called Ediperina. And that is a group of flies that they had studied them in Washington before, but we didn't know the extent of them and how far they spread. They were pretty sure that they laid eggs exclusively in barnacles, and all the records we had did. And we found larvae inside barnacles, we found pupa, we found adults crawling on them. But at the same time, we also found, if you look in this upper right hand, that just kind of looks like a barnacle, but inside of it, there are larvae of other flies as well, midges, chironomids, which often you're found in like ponds. They kind of swarm around golf courses in your backyard. But there's a couple of species that lay their eggs in barnacles, which is weird. Other places too, they'll actually lay them in algae growing on rocks as well. They're not really that picky, but some of them are inside barnacles, and the barnacle closes up and protects the larva. And then they sit in there and just filter things out of the water. So when the tide goes out and it dries out, the barnacle closes in and protects it, and then the tide comes in and it opens up and they filter the water. The barnacle flies themselves, the Ediperina, actually eat the barnacle. So over time they eat all the barnacle inside and then they crawl to the next one. It's a very strange life cycle, and one of those things that no, insects have nothing to do with sea life. But there are, and this was a good example of that. And it included, we had records from Japan, and we're not really sure which species are there, and also how they spread along from California up to here and have different timing. Some of them are only in the winter, but in California, they're kind of year-round. And we're not entirely sure what time of year they come out, but it's pretty specific. Around here, if I go out to Gonzales Beach or Ogden Point, about the last week of March, the first week of April, you'll usually find them. But if I go out there now, you probably won't find any adults. But if I go further north, when you get to Haida Gwaii, you can still find them in May and June. Alaska, maybe till July. Things we find out by looking at lots and lots and lots of records. And other things that aren't even insects, I said arthropod biodiversity, and these aren't all arthropods. There are hydroids, which are not arthropods, which Henry will study as well. But also a great group, and I'd love to have like videos of them moving, but they're so tiny. If you look on the left there, those are tardigrades, which are some of the smallest living things you'll ever find. They're called water bears, which is great because if you ever see, and actually some of the best ones are computer simulations of them because they're so tiny it's hard to get footage of them. But they do look like little 8-legged teddy bears that crawl around, and they sort of have a head, but sort of not, and they just sort of grab things and put them in their mouths, and they're very, very cute. They kind of look like marshmallows or teddy bears, but they're super tiny, and also strange in that they can completely dry out into a powder for years. And then you put water on them, and then they hatch out, and they keep going. So as a result, people know very, very little about them. They're so small, nobody ever collects them. They don't know what habitat to look in to find them. But Henry got in contact with folks from Europe, and he said, you know what, just grab any bit of dry moss or wet moss anywhere you go. So Henry started doing this, and pretty much every time we go, he finds a new species of tardigrades because no one ever studies them. And when you finally are able to extract them, and they're super tiny, and look at them, they will have all these features and be like, yep, totally different species, never seen it before. So this happens all the time. If you pick a group nobody works on, you can find new species. Don't pick mosquitoes. We know about mosquitoes. And the next thing Henry and I wanted to work on was rack specific. Rack, if you're not familiar with this term, is just dried seaweed. Technically anything that washes up on the beach is rack, and we look at the algae rack. Although it's not just algae, because you also get eelgrass in there, which is a plant. But you end up with these masses of things, and what we were finding is there are a lot of insects that are specific to that. And we started to look globally and be like, okay, what have people reported in other parts of the world? And there wasn't a lot of it. A few big studies in New Zealand, one big one that went across the coast of Namibia and down into South Africa. And then everything else has been kind of spotty. Like someone has a particularly favorite beach in Spain or in California, and they'll study what's on that one. But no one had really pulled it together, and certainly hadn't done it for British Columbia. So Henry and I started doing this. Like, okay, what do we have? I've got Henry's help to be able to describe the geology of the beach, the history, but also the algae that's on the beach. And then I can look at the flies that are there. So this was the most recent paper that we did. And we just looked at it and said, can you draw any conclusions about what's on which beach? Are they specific? And you can't be. It's funny, we were debating this over lunch today. You can never be 100% specific and say, this fly absolutely must be on this type of algae and it'll never be found anywhere else. It's never quite like that. It's more like 90% of the time it is found on a beach that also has this type of algae. And only once out of 84 visits did we ever find it on a beach that didn't have that algae. So that's why we chose to put it in terms of associations with algae type. We also found associations with substrate. That fly down there in the left corner, that is a fly. It's got two little stumpy wings, that's as big as they ever grow, and they can't fly. And it runs around on sand. And it's only ever on sandy beaches. And the weird thing is, even if we found a beach that is, you know, some of the beaches here that are just basically not slate, but big rock beaches, and there's not much, it's just rock. But you'll find little pockets of sand in places. You'll find those in the pockets of sand. But if you have a beach with absolutely no sand, then they're not there. Or if it's really big pebbles and no sand, they're not there. So it was associated with a very specific substrate. And then things like that at the top, those are kelp flies. They'll only be on a beach that has either Nereocystis or Macrocystis kelp in some way on the beach at some time of the year, then you'll find those. So we were able to do this with 33 different species of flies, and this was sort of a culmination of, 6-7 years of collecting over 84 different locations. And this was a case, it's like, no, this is what Henry and I collected, but then a lot of literature, you dive into the background to see what else is related. Although that's going long, so I should probably wrap it up there. But I'm happy to take more questions and go into more details if you want to, but I thought I would go there. Thank you.